Picture a quiet museum storeroom, metal shelves, cardboard boxes, a handwritten label that reads Rattus macleari, Christmas Island. The animal behind that name vanished from the wild just over a century ago, likely wiped out by diseases that hitched a ride from ship rats. Now, those dried skins have become a test case for something that sounds like science fiction, rebuilding an extinct genome, and one day, maybe the animal itself. Scientists managed to rebuild most of the Christmas Island rat's genome from those old skins. On the surface, that sounds like revival. A recent extinction, a close living cousin with a gold standard lab genome, and modern tools doing the heavy lifting. But tucked inside the data is one small worrying word that changes the whole story. Almost. Some gene blocks are still missing, and a lot of the weak spots land in smell and immunity, small gaps with big jobs. 95% sounds comforting until you remember you wouldn't board a plane that was 95% assembled. If you're new here, welcome. If you're one of the wild ones, you already know the rule. Receipts first, verdict later. In this episode, we take the neat, hopeful headline and stress test it. How best case is this really, when the closest relative is a fully sequenced lab rat and the DNA is only about 100 years old? And if 95% of a genome still misses the genes that keep a rat alive in the real world, what does that say about bringing any extinct species back at all? The pitch sounds perfect. Started with an island rat that went extinct only about 100 years ago. Its closest living cousin, the Norway brown rat, already has a gold standard reference genome. Their last common ancestor is only a couple of million years back, which is very close by evolution standards. Take tiny samples from 2 museum skins, run the ancient DNA through modern sequences, and map every fragment you can onto the brown rat reference. The result? About 95% of the Christmas Island rat genome can be reconstructed at high coverage. On paper, it looks like a comeback in progress. But the team behind the study did not stop at the percentage. They asked what lives inside the missing slice. When they dug in, they found that nearly 5% of the genome was unrecoverable at all. Over 1,600 genes were under 90% complete, and a smaller set of genes was missing entirely. Those gaps were not random noise. Genes linked to immune responses and smell were overrepresented in the weak or missing group, exactly the tools a wild rat uses to find food, dodge danger, and cope with parasites. So the tidy claim of 95% recovered hides a much heavier lift. The most fragile, behaviour-shaping parts of the genome are some of the first to fall when you rebuild a species from fragments. As a proof of concept, this is our first quiet red cross. So what do we actually have right now? No frozen Christmas Island rat tissues, no cell lines on standby, no Jurassic Park vial waiting to thaw. The last real animals died out on Christmas Island in the early 1900s. What researchers worked with were 2 taxidermy skins more than a century old, stored in the Oxford University Museum of Natural History. From those skins, they pushed the sequencing as far as current tools allow and mapped the fragments onto the brown rat reference genome. That is where the famous 95% figure comes from. At first, that number sounds reassuring, almost complete. But once you know that almost 5% of the genome is unrecoverable and that many smell and immune genes live inside that missing slice, the picture changes. What we hold today is not a plug-and-play digital clone of Rattus macleari. It is a mostly complete blueprint with some of the most important pages blurred or ripped out. Impressive as a genome milestone, absolutely, but it is still information on a screen, not a living rat, and the missing functions are exactly the ones you need if you ever want this story to leave the lab. Even if you treat the genome as good enough, a Christmas Island rat will still have to grow inside a living body, and that body will not be extinct. In practice, the plan would be to edit the germ cells of modern rats, almost certainly brown or black rats that already breed well in labs. Those animals would carry edited eggs and sperm. Their offspring over several generations would be pushed closer and closer to the target genome. Technically, that is attractive. Rats are small, reproduce quickly, and there are established protocols for hormone treatments, embryo transfers, and surgeries. But each neat arrow in a gene editing diagram translates into real procedures on real animals. Many surrogates would go through repeated cycles of treatment, surgery, and culling to troubleshoot the pipeline. The welfare bill rises with every failed attempt that never makes it into a press release. Then layer on the immune system problem we already met. The original Christmas Island rat likely went extinct in part because of introduced disease. If you somehow restored its exact old immune profile, you might simply rebuild an animal that is unusually vulnerable to modern pathogens. If instead you quietly patch in more robust modern immune genes from lab rats to keep it healthy, you slide one more step away from resurrecting the old species and one step closer to designing a hybrid that only wears its name. On surrogate risk and welfare alone, this does not look like a straightforward green light. Suppose the lab hurdles are cleared and you end up with a small breeding line of animals labelled Christmas Island rat. Where do they live? The obvious answer is in the name. They evolved on Christmas Island, a small forested Australian territory in the Indian Ocean. When they were common, they probably helped control some of the other species, including the famous red land crabs. If you take anything away from this, let it be this. Losing the Christmas Island rat may be one reason the red crab migration is such a massive world-famous spectacle today. And really, if you're booking a holiday, are you going for a crab migration or a rat migration? Modern Christmas Island is not frozen in that moment. It has ports, rubbish tips, houses, and public health rules, plus more than a century of work trying to limit rodent-borne disease. Any serious reintroduction would mean explaining to residents why scientists now want to ship in protected rats and then pay to monitor them for decades. You would need biosecurity, health screening, long-term funding, and a plan for what happens when some of those rats raid kitchens, grain stores, or rubbish heaps. At the same time, conservation budgets are finite. For the cost of building and managing a resurrected rat on one island, you could protect habitats and tighten biosecurity for multiple living rodent and small mammal species that are sliding toward extinction right now. From an ecosystem first view, this project starts to look less like repair and more like an expensive science experiment with a very narrow payoff. The Christmas Island rat is a clean reminder that genome percentages can be deeply misleading. 95% sounds like an A grade, but what matters is what lives inside that missing 5. In this case, the team found that genes tied to things like coat and whisker traits were recovered pretty well, which means you can probably get the look more or less right. The weak spots were in genes linked to smell and immunity, systems that decide how a rat senses its world and how it holds its own against germs. Think of it like restoring a laptop from an old backup. You might get almost all your photos, music, and desktop layout back. It feels familiar, but if the missing files include the network drivers and security updates, the machine is going to struggle the moment you connect it to the real internet. With an animal, losing or guessing key immune and sensory genes is the same kind of quiet failure. The body might look right, but the behaviour and survival odds are way off in ways you cannot see in a still image. In plain language, you can't just count how much DNA you've rebuilt. You have to ask, did we save the parts that actually keep this animal alive? If you need a quick comparison, use this one. Calling that a comeback is like saying a plane with 95% of its parts is ready to fly. Pity the missing bits are the wings and the landing gear. A high percentage with broken functions is not a win. On the core question of whether almost genetic recovery is good enough for a true comeback, the Christmas Island rat sends a clear warning printed in red. One more big question sits in the background of this whole story. What are we trying to achieve that we cannot get any other way? With huge mammals like mammoths, people argue there is no perfect stand-in alive today. With rats, that is not true. Brown rats, black rats, and other close cousins already fill very similar ecological roles on islands and coasts all over the world, sometimes too well. If your scientific goal is to understand how a native rat once behaved or how it shaped an island food web, you can learn a lot from models, simulations, and living relatives without building a custom hybrid. From a conservation point of view, the trade-offs get even sharper. Many small mammals and island rodents are in real trouble right now. Their genomes are intact, their behaviour fits their ecosystems, and they are losing ground to habitat destruction, poisoning, and invasive predators. Steer the same money and lab effort into protecting them, and you keep whole branches of the tree of life from snapping off in the first place. Protecting dozens of real species beats resurrecting one uncertain ghost. It is the smarter flex, the calmer story, and the kinder long-term move. When you stack up proxies and prevention against the Christmas Island rat de-extinction plan, the option that looks most like a true green light is not the one with CRISPR in the thumbnail. Put it all together, and this really is a best-case stress test for gene editing de-extinction. The Christmas Island rat died out only about a century ago. Its closest living cousin, the brown rat, has an excellent reference genome and is already a lab workhorse. Researchers squeezed as much information as possible out of the two museum skins and managed to recover around 95% of the genome. That is a real scientific achievement and a clear sign of how far ancient DNA work has come. But none of that erases the cracks we have walked through. Almost 5% of the genome remains unrecoverable. More than 1,600 genes are incomplete and a smaller set is missing outright. Many of the weak or absent genes hit smell and immune functions, which shape how a rat survives and behaves in a real environment. Any animal grown from that blueprint would either carry fragile, reconstructed versions of those systems or patched in modern lab rat versions that quietly push it away from the original species. Surrogate rats would pay a welfare bill to test each step. Christmas Island itself has changed, with people and policies that treat rats as health risks, not treasures. And existing threatened rodents could be helped far more directly with the same money and time. So the verdict here is not whether the work is clever. It is. The blunt line is different. Genome does not equal animal. A 95% digital copy with missing core functions is not enough for a true comeback. For Christmas Island rat de-extinction, with today's tools and today's priorities, my verdict is veto. Genome does not equal animal. If this rat has a lesson for the whole de-extinction conversation, it is this. The hard questions hide behind the shiny numbers. How recent is the DNA? Which genes are gone? How many living animals carry the risk? What kind of home waits at the other end? And what else could we be doing with the same resources? Wild ones, this is where I want your head to stay. When you see a headline about bringing something back, look past the percentage and ask those same questions. In the next episode of this Greenlight or Veto series, we will take that same checklist to another candidate and press just as hard. Is this really a path toward repairing what we broke, or just an expensive way to build hybrids that only look right at a glance? As always, we will start with the receipts and end with the verdict. If this breakdown helped you see past the hype, hit like, subscribe, and drop your verdict on the Christmas Island rat in the comments. Would you call this a prototype, a partial success, or a hard no? Now, here is another video I know you will love. I'll see you over there, and thanks for being one of the wild ones in this series.